we're here with the cell cycle. We are moving into how cells go about dividing, and they have to go through a series of steps before they are able to grow and divide. And understanding this cell cycle will help us better understand how cell division is taking place and what are the necessary precursors for that to be able to have a successful cell division, right? So let's look more closely at what will be the cell cycle here. We are going to represent it by a circle, and a majority of the time, cells are in this phase called interphase. And interphase is going to be comprised of what will be three more phases, the G1, the S, and the G2. Now, in G1, so we have the start, we are going to need to double the amount of organelles we are starting with. So when we split, our new cell or identical sister cell will have all of the necessary organelles to go about its processes that is going to be responsible for. So in this case, we are going to make organelles. We can also make proteins here. And the majority of a cell's life is spent in this phase, right? Or it's spent in this step of the cell cycle. Once we have double of everything, we can move to the S phase. S stands for synthesis. So in this, we are replicating the amount of DNA we have. So I will say that we are doubling the amount of DNA we have. So if we have 23 pairs, we will be doubling that to what is 46 pairs. So now while we go through and divide, once we split, our new cell will have 23 pairs of chromosomes. Once we've gone through making more organelles, we've now doubled our DNA, and we can go to the last step, which is the G2. In this, we are preparing for mitosis, and we are checking, right? So we are going to check for everything being set. So we are going to make sure that all of our DNA is ready to go. We are going to make sure that we have every organelle that is doubled and ready to go and move on. We are checking to make sure that we're at the right size even, right? We need to also grow in the amount of size we have, and that is kind of consequence of making more organelles, right? So this is all interphase, and this phase is roughly somewhere about 22 hours long. So a majority of life will be spent here in this phase. Now let's say that we aren't ready to grow, or we are a very highly specialized cell that maybe we don't need, so we have no need for cell division. Well, then we can enter what is kind of this holding state known as the G0 or the G0 phase. And good examples of this are things like your neurons, right? So when we say you're losing brain cells, well, they don't regenerate, or we're not actively making more and more neurons or brain cells constantly throughout our lives. It's very difficult. They're very highly specialized, and our DNA just doesn't have that. So we can go through interphase. we can double the amount of DNA, we're doubling the amount of organelles, we're checking everything, and if that's all good to go and we get the green light, then we can move into the final phase, which is going to be called the M phase. And this M stands for mitosis. And within here will be a series of steps that we go through. And in here, this is where actual cell division takes place. So let me call this what is going to be interphase, and then we lead into what will be actively cell division. And this will take approximately 2 hours of time. We are dividing our cells here, so I can maybe place a cell divides, and we are essentially taking one cell and splitting it into 2 cells. So that's going to be very helpful when we need to repair lost cells, when we need to make new cells, and so forth and so on. All right, thanks for watching. Stay tuned.